In this video, we will study about messenger RNA, also called as mRNA. RNA is ribose nucleic acid. This term was coined by scientists Jacob and Monod in 1961. In a cell, total RNA is made up of messenger RNA, ribosomal RNA, and transfer RNA. Out of the total RNA, messenger RNA constitutes 2 to 5%. And when we centrifugate it, it can sediment at the base during centrifugation, and we can calculate its coefficient, that is called as sedimentation coefficient. And sedimentation coefficient of messenger RNA is 6 to 30s. As the name indicate, messenger mean this will carry the messages. So this is called as messenger RNA. This carries the messages from the DNA and helps in formation of the protein. Whatever information is present in the DNA, that will be translated in the form of protein. So this is called as messenger RNA. So we can say messenger RNA carries the protein blueprint from our cell's DNA to its ribosomes. Now, how this occurs, we can understand by this example. Now, this is DNA. This is double stranded DNA. One is 5 prime to 3 prime, another is 3 prime to 5 prime. One is template, second is non-template. Now, template strand will help in formation of messenger RNA. Now, this is the template strand. It is having sequence AGA, AGT, TTA, CTA, CCA, GCA. This I have made hypothetical sequence just to make you understand. And now these are called as triplet codon like AGA, AGT. These are triplet codons. Now they will code for amino acids. Now when from DNA, messenger RNA is produced, now its sequence will be complementary to this sequence or opposite to this sequence. Now A will pair with U, G will pair with C, and A will pair with G. Then again, opposite to A is U, opposite to G is C, opposite to T is A. Because in case of RNA, thymine is not there, it is replaced by uracil. So opposite to A it will be U. So similarly, opposite to TTA it will be AAU. And opposite to CTA it will be GAU. So these codons, which are complementary to the codons present in the DNA, now they will be converted into amino acids by the process called as translation. So messenger RNA, ribosomal RNA, and transfer RNA helps in formation of polypeptide chain. So this formation of protein from the DNA is called as translation. Formation of RNA is called as transcription. Now UCU will form serine, UCA will form proline, AAU will form asparagine, GAU will form aspartic acid, GGU will form glycine, CGU will form arginine. So polypeptide chain will be produced. So we can say genetic information was present in the DNA that has been translated in the form of a polypeptide chain or protein. Now if we study the structural component of messenger RNA, see its structure. What is its structure? Now this part is called as cap region and this part is called as tail region. Now in the nucleus from the DNA, messenger RNA is produced. Now this is the messenger RNA which is being produced from the DNA. Now when this comes out into the cytoplasm, before coming out, a cap will be added and tail will be added. Now this will cap and tail will protect the messenger RNA in the cytoplasm from the action of the enzymes. Now this is the cap part which is present at the 5 prime end, and this is the tail part which is present at the 3 prime end. Now this part is called as non-coding region 1. This is non-coding region 2. Non-coding as the name indicating, this will not code for any of the amino acid. So this will not form any protein part. And this part is called as coding region because it is going to form the protein part. And this part is called as initiating codon because this will help in formation of the polypeptide formation. And this is called as termination codon because this will help in termination or stopping of the polypeptide chain formation. Now this non-coding region 1 is also called as leader region. Non-coding region 2 is also called as trailer region. Now we'll study one by one all the parts. Now this is the cap part which is present at the 5 prime end. Now this is highly methylated This will protect the messenger RNA from action of the enzymes. This will prevent its degradation. And this part will also help in binding of the messenger RNA to the ribosomes. Like ribosomes, they bind to the messenger RNA. So these are the codons which are present on the messenger RNA, and this is the ribosome. So this messenger RNA, its cap region, will help in binding of the messenger RNA to the ribosomes. Now next is non-coding region 1, which is not going to code for any protein. This is made up of 10 to 100 nucleotides, and this region is mostly made up of A and U bases, and this is also called as leader region. Now this part, as we have discussed, this is initiating codon. This part is the coding region. So in the initiating codon, we can say this is the first codon which will form methionine, help in initiation of the protein formation. So this is AUG, this will code for methionine. GUG will code for valine. Now next is coding region. This is made up of 1500 nucleotide. Like this part is the coding region, and this will be having codons like this, and these codons will form amino acid like we earlier discussed. Now this is amino acid 1, 2, 3, 4, any name it can be produced, AA5, AA6. Now they will combine to form polypeptide chain. So this is the polypeptide chain which is being produced from the coding region. And when this will reach toward the termination part, this is the termination part, like here it can be UAA, UAG or UGA, any of the triplet codon will be present at this end, and this will, like all these codon, they are recognized by the transfer RNA. This is the transfer RNA which is carrying the amino acid, but here it will not be recognized any of the transfer RNA. So here when it is not recognized, these are called as stop signals or nonsense codons. So these are called as termination codon, which helps in termination of the protein formation. So they can be UAA, UAG, UGA, which are also called as nonsense or stop codon, because they help in stopping the protein formation or polypeptide chain formation. Now next is non-coding region 2. This is made up of 50 to 150 nucleotides. This is made up of AAU, AAA sequence. This is also called as trailer region. Now at the end of the messenger RNA is present poly A sequence. This is made up of 2 to 250 nucleotides. Sometime this may be made up of less nucleotides. So cap and tail, they protect the messenger RNA from the action of the enzymes, as well as they help in binding of the messenger RNA to the ribosomes while protein synthesis. Now if messenger RNA forms single polypeptide chain, this is called as monocistronic, and this is very common in case of eukaryotes. But if messenger RNA forms more than one polypeptide chain, many polypeptide chain, this is called as polycistronic, and this is common in prokaryotes. Now messenger RNA, as we know, this will get degraded very easily with the help of enzyme. So that is called as half-life of messenger RNA. It may remain in the cell 1 hour to several hours. Now this is acted by enzymes ribonucleases, but when this messenger RNA forms complexes with protein, this may be protected from the action of the ribonucleases which degrade the RNA. Then it forms a stable structure called as informosome. What is informosome? Complexing of messenger RNA with the proteins. So we can say primary function of messenger RNA is to act as an intermediary between the genetic information in the DNA and the amino acid sequence of the protein. Like we have already seen, now this is the template strand of the DNA. Now this will form messenger RNA, and messenger RNA will form polypeptide chain, that is protein formation. So whatever information is present, genetic information is present here, or hereditary information is present here, that will be carried by messenger RNA in the form of polypeptide chain. This is called as transcription, and this process is called as translation. Thank you for watching my video. If you like my video, please like, share and subscribe.